Hello, everyone, and welcome to today's webinar, Memory and the Brain. I'm Manuella Yassa, Director of Outreach and Education at the University of California, Irvine's Center for the Neurobiology of Learning and Memory. I'll be moderating the webinar today. During this presentation, you'll hear from myself and my colleagues for this at-home Brain Awareness Week event, where you can participate in hands-on activities and learn about memory and the brain. We'll address questions from the audience at the end of the presentation. Now, let's get started. I have a great team with me today. Unfortunately, Jessica Bolton wasn't able to make it, but I do have Rianne and Alberto, who are both graduate students, working here at UC Irvine with Dr. Marcelo Wood. They're working on earning their doctoral degrees. Today we're going to be talking about memory and the brain. What is memory and how does it work? We're going to tell you the story of HM, who was a very famous patient who had some memory problems, and he taught us a lot about memory. We'll also be taking your questions. You're welcome to ask questions throughout the session. However, we will wait until the end to answer them. To start off, let's make sure everybody is on and live and is able to answer questions. So my first question for you is, where are you all tuning in from today? Are you from the US? Maybe you're from the West, Midwest, South, or Northeast? Or maybe you're tuning in from somewhere else. So I'll give you a couple of seconds to answer. Oh, it looks like we have some answers coming in. Fantastic. We have some folks from the Midwest, some from the Northeast. Anybody from the South? Oh, some people from the South. Fantastic. And more actually tuning in from the West. We may be the only ones. We have some people from tuning in internationally. I'll give it a couple more seconds and then I'll show you the results. It may be too early for those of us in the West. Fantastic. So we're going to move on. Here we go. We have folks from all over the place. Beautiful. So that's how you'll be answering questions throughout the webinar. Now I'm going to turn it over to Alberto, who is going to start talking to you about memory.
Good morning, everyone. This is Alberto, or at least it's morning for us here in California. So before we can talk about how memory works, we first have to get a strong definition for what memory is. So let's start by thinking about the things that we have to remember every day. So we'll start with this simple exercise of answering these simple questions. So how old are you? I'm 29, so that's a thought that keeps me up at night. What did you eat for breakfast this morning? What's your favorite food? What's 2 plus 2? What's the name of your school? And how do you throw a ball? So these are questions that we have to know the answers to pretty much every day. But let's see if you could name these individuals. So this is obviously Mickey Mouse. Disney's pretty close to us. This is Dr. Seuss's Cat in the Hat. And this is Anna from Frozen. And so hopefully you remember those people. If not, we'll see. And so now that we kind of get this idea of being able to answer these questions, these facts that you need to know every day, and recognizing everyday objects, we'll really see when it becomes important to use that information. And so let's see if you can tell these two items apart. These two objects look very similar, but have very different functions, right? So they both kind of look the same. They both have a handle. They're both blue and white. They're both bristly. But it's really important that you recognize that one is a toilet brush and the other is a toothbrush, right? So if you weren't able to tell that difference, you might end up putting toothpaste on that toilet brush and pretty much ruin the rest of your day. You would not want your day to start that way. But it's also important to remember that some memories can be tied to very strong emotions, and simply by accessing that memory, they can make you feel a specific way. So here we have two different animals. One is a cute little puppy. The other is a pretty gross spider. And at least for me, the puppy makes me feel warm, happy, reminds me of my childhood dog, whereas a spider makes me feel scared and anxious, and it reminds me of the last time I got bit by a spider. So it seems, we can put all this these slides and information together to generate this idea that memory is your personality, your talents, your skills, your habits, your life story, your preferences. Essentially everything that makes you you can be summed up as this kind of mosaic of memory. And we are who we are because we can learn and remember new skills and new facts. So what would happen if we couldn't remember? And so for that story, I'll pass it over to Rianne.
Hi, everyone. So we're going to tell you today about the story of a famous person in the world of neuroscience called Henry Molaison, or HM. And so HM was born in 1926, and as a child he began having epileptic seizures. The epileptic seizures are when you have an overactive or hyperexcitable brain state. So you might wonder what that really means. Well, first we should really talk about the basic units of that make up your brain called cells. See, cells in the brain are very different from other cells in the body. They're special because they can talk to each other in ways that other cells can't. See, brain cells are called neurons that use both electricity and chemicals to communicate. And so here's an example of how they do this. What we have here are two types of neurons. One is sending the signal, and the other is receiving that signal. This sending neuron sends an electrical signal down its long projection that can trigger a release of chemicals into a space between the neurons as a way to communicate to the other cell. That space is called the synapse. These chemical messengers travel across the synapse to attach to the receiving neuron. When those chemicals attach to the second neuron, it produces electricity to send this message then to the next neuron. So, as our brain has cells that use both electricity and chemistry to communicate important messages to each other, that's how it keeps our brain working. So what does this have to do with HM? Well, we need just the right amount of electricity to have our brain working properly, right? Well, if you have too much electricity, you can have a seizure disorder. That's what started happening to HM when he was 9 years old. And seizure disorders can damage the brain. So doctors tried to stop HM seizures with medication. Unfortunately, HM seizures were so bad that medicine could not stop the seizures from happening. So when HM was 27 years old, Dr. William Scoville found that his seizures were starting in the part of the brain called the medial temporal lobe. Do you want to know where the medial temporal lobes are located in the brain? Well, I'm going to turn it over to Manuela, who will take you a little tour of the 3D brain, which you can explore at home.
A really great place to explore the brain is on BrainFacts.org. They've recently launched the 3D brain, which can be found at BrainFacts.org/3dbrain. I'm really excited about this, and I use it a ton because you can actually play with the brain and explore the different structures. So here I am, I'm spinning the brain around. If I go like this, the brain will be looking to the left side. So the eyes would be here. The back of the head is here. Down here we have the cerebellum and the brain stem. When you hover on a structure, it shows you the name of the structure, which is fantastic. The part of the brain that we're interested in today is the temporal lobes. The temporal lobes sit on the side of the brain, and to see it more clearly, I'm able to go here and go down to temporal lobe. And when I do that, the temporal lobes will be colored. So if I spin the brain this way, I can look at the bottom of the brain, which is a really interesting view because it shows me the temporal lobes very clearly. Here we can spin it back around so that you can see the side. So the temporal lobes sit right next to your ears. And there's one on each side. Now inside the temporal lobes there's a really interesting structure called the hippocampus, which you'll hear more about today. If we go here, and go down to hippocampus, you'll be able to see that structure more clearly. It's a really neat structure and kind of a uniquely shaped structure. So this is the part of the brain that Dr. Scoville removed during HM's surgery, and you'll learn more about that in a little bit. I hope that you'll take the time later this week to explore the brain on BrainFacts.org.
Okay, so now that we know where the medial temporal lobe is located, once Dr. Scoville realized that that's where HM seizures were coming from, he performed a surgery to remove those areas, hoping that it would then stop his seizures. So on the left here we have a few images to show you of what it will look like if the medial temporal lobe was removed. So we have the normal brain on the left, which is where, and you can see on the left is where the eyes are usually located, and on the back of the right is the back of the brain. In order to see the inside of the medial temporal lobe, we now removed a part of the brain in order to make this visible for you. See, the bottom left image shows the temporal lobes at the bottom of the brain. On the right side, we have HM's brain after the surgery. As you can see, there are big holes where the temporal lobe should be. And so after Dr. Scoville removed those parts of the brain, he noticed that HM was having major memory problems. And HM could not remember conversations that he had with hospital staff and kept introducing himself to the same person multiple times. When Dr. Scoville realized that that was happening, he called up Dr. Wilder Penfield and Dr. Brenda Milner from Montreal to help assess HM's memory. Here is a picture of Dr. Brenda Milner, who is considered a very important scientist in the world of learning and memory research. This is a more recent picture of Dr. Milner that was taken last year by the New York Times. Dr. Milner is now 99 years old and is still as curious as ever. She recently said, "People think because I'm 98 years old, I must be retired. Well, not at all. I'm still nosy, curious." So, being the curious scientist that she is, when Dr. Milner heard about HM's memory problems, she packed up her neuropsychological tests and went to find out what might be happening. So, once Dr. Milner met HM, she conducted some tests. One of the tests she used is called the words list test. Let's see if you can do one of these tests now that Dr. Milner used on HM. So, I'm going to give you a list of words, and let's see if you're able to remember them. So don't write these down. Here are the words: window, dog, flower, girl, feather, spoon, apple, car, bed, and sand.
Hey, Rianne, let's show everyone where we're tuning in from.
Sure, Manuela. I guess we can switch gears for a couple minutes.
Okay, everyone. So everyone's tuning in from all over the world. So let's tell you where we are. So here we're going to zoom into North America. We have the United States. And then we zoom into California in the western part of the country. If we zoom in a little bit more, you'll be able to see Los Angeles and San Diego. We're going to zoom in right in between those two cities to Orange County. Many of you will recognize Anaheim up there. That's where Disneyland is. We have Huntington Beach and San Clemente. And we have right in the middle there a place called Irvine. So if you zoom into Irvine, you'll be able to see the University of California, Irvine, which is where we are. And a little bit closer, you'll see Aldridge Park in the middle, and to the left of Aldridge Park is our building. And here we are, all four of us, standing under a picture of Dr. Milner. Okay, Rianne, I think we've given them enough time to distract them enough so that their memories can get into their hippocampus. Let's see how they do.
All right, so let's see whether or not you're able to remember the words I gave you before. So here are a list of words that I'm going to show you, and you tell me if you've seen them before. Bus, pink, flower, plant, leash, spoon, door, bed, sand, boy. All right, so how did you do? Which ones do you remember? So on the left are the words that I gave you previously. And so on the test, we actually have highlighted words that we didn't give you before that are kind of considered distractors. You might notice that they're a little similar to some of the words that we gave you before. And I mean, memory's pretty complex, and so we can go over later why you might have misremembered that because they're so similar. But for now, let's see, how did HM do on this test? Well, it turns out he couldn't remember any of the words that he was given on that list. He didn't even know that he was given a list. So why is that? Well, let's go back to the surgery to really understand this better. So earlier, I told you that Dr. Scoville removed a part of the brain called the medial temporal lobe. Well, it turns out that one of the pieces of the medial temporal lobe is a structure called the hippocampus. You can see that the hippocampus is actually shaped like a seahorse. It was named the hippocampus because it's actually Latin for seahorse. And so when Dr. Scoville removed HM's hippocampus, it's actually no longer able to create new memories. This is called anterograde amnesia. So the hippocampus is necessary for this type of learning required to memorize a list of words. And so this type of learning is called declarative. Declarative learning, or learning new declarative information. And so this is the kind of information that you can recall or declare, such as facts, events, dates, or faces. So we found out the hippocampus is necessary for this kind of learning, but is it necessary for all types of learning? So Dr. Milner began to wonder, can HM learn anything new? Well, Alberto's going to take us through the next test that Dr. Milner used to find this out.
Hello again. So Dr. Milner asked, can HM learn a new skill, right, which is a little bit different than learning a new fact. And so to test that, she used the mirror drawing task. So we're going to go through kind of the logistics and how to set up the mirror drawing task today. So the idea is that you place a star in front of a mirror, and only looking through the mirror, you try to trace the star while staying in between the lines. And so here's an example of how it would look like with a perfect score, where you start with the S, and you trace through the lines without making any errors, crossing through the double lines. So for those of you who are trying to do this at home, we can set you up. We're going to walk you through the setup, and what you'll need is a mirror, or the mirror drawing task printout. So that's the image with the double star, an actual mirror, a piece of cardboard or a piece of paper to block your view of your hand so that the only way you can see the paper and your hand is through the mirror. You'll need a pencil for doing the task and counting your results, a clock or a stopwatch, something to see how long it takes you to do the task. And then optional is a piece of tape to hold the paper in place while you're tracing. So now I'm going to demonstrate the task for you. This was the first time I had ever done it, so we'll see how I did. Hello again, I'm Alberto, and today we're just going to show you how the mirror tracing task works. So essentially to get it going is you need a marker, a mirror, something to block your view, and then the actual thing you're going to trace. Here we're going to be tracing a star today. And so the important thing is when you set up is that you're blocking your view of your hand, and the only way you can see your hand, the pen, and the paper is through the mirror. And so this is the first time I've ever done this, so we'll see how it goes. Part of me is worried that I'm going to do badly. The other part's worried I'm going to do horrendously. So we'll see how it goes. Okay, so there's a start point at the top. And obviously, because it's a mirror, things aren't necessarily working. Your hand isn't going to necessarily work the way you think it's going to work. And the whole point is to try to keep the tracing in the line and try to do it as fast as you can. You can already see I'm starting to waver a little bit. That one's getting a little easier. Oh, there, things are coming off the rails a little bit. Okay, almost done.
Done.
Okay, so that was a little bit harder than I thought it was going to be. But so for those of you who are trying to do this at home, I'm going to walk you through the instructions one more time and give you time to set up and try to do the task yourselves. So to start, you put your piece of paper in front of the mirror. You put your cardboard or whatever you're using to block your view between you and the paper. You look into the mirror and make sure you can only see your hand and the star through the mirror. So if you are cheating a little bit and can see your actual hand, kind of shift where you are, shift where your paper blocker is. And so then starting at the S, start tracing your star. Hopefully you can stay between the lines a little bit better than I did. And have your partner time how long it takes you to go all the way around. And once you're done, count how many times your pencil touched the edges of the path. So not just how many times it crossed it, but how many times it touched it. And count up your findings, and then we can compare your results. So how did everybody do? Chances are your star does not look like the one on the left, but it looks more like this one on the right, where there's a lot of zigzags. It's really hard to keep track of where your hand wants to go, where your hand needs to go, and what your hand actually does. So this kind of star is something that looks like what I would trace, what you would trace. But the question is, would HM trace a star like this? So when Dr. Milner tried this task with HM, it also looked like this when he started. So then we're going to go through some of our samples. I'm not going to mention any names, but it wasn't Rianne who drew these. And you can see, so at the first trial, the person doing this really had trouble getting past just that first corner. But then she was really struggling. But then the second time, she got much better. She was able to go all the way around. But you could still see at corners there was some issues getting your hand to turn in a different direction with the mirror. And then by the third trial, she did pretty good. She did pretty well. She was able to go all the way around with pretty few errors. And so if we were to do this at home, what happens after several trials? So you'd want to keep track of how well you were doing over repeated trials. And you could even do multiple trials in one day. And so here you can see on the left, keeping track of the number of times you've done it, so the first, the second, the third, or the fourth. And then on the right, keeping track of how many mistakes you've had at each trial. And so if we were to graph this out where the bottom is the trial number, so from left to right, this is what I did on the first one, the second one, the third one. And then on the left axis, the y-axis, keeping track of how many mistakes you had, you expect at the first trial to have a lot of mistakes, so 27. But then as you go through it, you see that you have less and less and less mistakes. So it seems like something's happening where each time you go through this, you have fewer and fewer mistakes until you get to the 10th trial where you're almost able to do it perfectly. And so there's clearly a trend. So as the number of trials increases, the number of errors decrease, or you get better at the task. It gets easier because you kind of figure out the best way to use the mirror to actually get your hand to do what you want it to do. So that's my data. So clearly I'm learning how to do this task. Hopefully when you do this at home with your parents or with your friends, you all will learn how to do this task also. But what about HM? We've already talked about him having a memory problem. Can he learn how to do this task? So did HM's data look like this? That's what okay, we'll let you answer this first. Take a guess.
So to remind you, the question is this what HM's data looked like, that downward slope? Oh, we have some answers here. Let's see. Seems like some people think that yes, this is what HM's data looked like, and some people think that no, this is not what HM's data looked like. We'll give you a couple more seconds to respond, and then Alberto will tell you more.
Okay, so it seems everyone's had a chance to answer, and it seems like most of you think that HM's data did not look like this, so he did not get better at the mirror tracing task. Well, Dr. Milner had this question also, and so she kept track of his data, and you can see through his first star, which is at the top, that he also seemed to have the same problems that not Rianne had during the mirror drawing task. But she also noticed that during that same day, he was getting better. He was making fewer and fewer mistakes, that by the third day, he had under 5 errors, which is even better than me. So clearly something's happening. Just looking at the data, we know HM's learning something. But Dr. Milner wanted to know, what are you learning? And so she asked him, have you done this task before? And so when she asked him, his response was, I don't think so. I don't recall. Which is crazy. He's clearly getting better at this task, but he doesn't remember ever doing it. How is that possible? And so from this data, we're able to learn that HM was not able to form new declarative memories, so that word task. So he wasn't able to learn things that you'll need to tell a story, right? The things that Rianne talked about. And so from that data, we can generally understand that you need the hippocampus to make new declarative memories. But from the mirror drawing task, HM was able to learn new skills. He clearly got better at the mirror drawing task. So it seems like the hippocampus is not required for skill learning. And so for all of this information, it seems pretty obvious to us now, but back in the 50s, we had no idea how these processes in the brain worked. So we have a lot to thank HM for. But his contributions to science didn't end there. So in 2008, unfortunately, HM passed away at 82. And the next year, he had donated his brain to science, and the next year it was sliced into what are called histological sections to create essentially HM's brain map. And so those black lines show the direction of the cuts. So those are called coronal sections. So those are essentially slices that are coming down from a crown. And those pictures there are actual pictures of HM's brain. And you can go at home and look at them yourselves at the Brain Observatory website that we have there. But you can actually go find and see the parts of the hippocampus in HM that are missing. And so to close up, I'm going to pass it back to Manuela.
So we've placed here a few resources for you, a link to the 3D brain, a link to HM's Brain Atlas. We've actually put in here also the link to the New York Times article about Dr. Brenda Milner. It's an interview with her, and it's fantastic. I urge you all to go check it out. And we are happy to take any of your questions at this point. So if you have any questions, you can go ahead and place them in the Ask a Question. And we'll give you all a few minutes to answer, or to ask, not to answer, to ask your questions.
Hi, so this is Alberto. So it seems someone asked, can memory be actively improved? And there's a lot of research going on throughout the country, throughout the United States, including here at UC Irvine especially, trying to actually understand how these cells that Rianne talked about change the way they behave to generate memory. And so the short answer for the can memory be improved, yes, definitely memory can be improved. How to improve memory becomes a little bit trickier. So early research, a lot of research has shown that one of the strongest ways to improve your memory is sleep. So everybody has seen the movies and TV shows where somebody forgot to study for their test and they're going to spend all night cramming for their exam. That's actually the worst thing you could do. Sleeping after you study is actually one of the best ways you can improve your memory. Also things like exercise and nutrition. It sounds fishy, but exercise actually has been shown to help you form new memories and keep those memories longer. So I'll pass to Rianne for the next question.
Hi, everyone. So we have another question here saying, what is the role of emotion in forming memories? And so some of you might realize when you're actually, when you have a lot of emotions, let's say maybe you are really upset or angry, you tend to remember those kind of experiences really well. You remember a fight and what someone really said if you're really upset with them. Or if you just got hit, maybe you really remember that incident. And so that one of the brain regions that's involved for this is not just the hippocampus, but actually the amygdala. And it turns out the amygdala is a brain region that kind of communicates with the hippocampus to kind of modulate and really heighten the memory storage of that type of memories.
Excellent. And on that same note, there's actually a very famous neuroscientist here at the center. His name is Dr. James L. McGaugh. He's the founder of the center here. And he spent his life career studying exactly that, how emotion modulates our memories. And so that's a really interesting topic, and research continues on that topic to this day and will continue for a very long time. Let's see, what other questions do we have here? One of the questions is, what types of fitness exercise can improve memory? And we're just starting to understand the relationship between exercise and the brain. And the simple question is really all types of fitness exercise. Anything that gets your heart pumping will be good for your brain. And in fact, anything that is good for your heart is good for your brain. So when we talk about nutrition, a lot of times people will say, well, you know, what can I eat? I hear that blueberries are good. I hear that, you know, avocados are good, or and really the answer is that anything that is good for your heart is going to be good for your brain. There is not a lot of data on the nutrition side right now, other than the Mediterranean diet, that is going to really, that there's data suggesting that is really going to improve your memory. However, there is a lot of research happening right now. So back to the exercise, it's really anything that gets your heart pumping and gets you sweating. And that might be different for a 15-year-old and for a 90-year-old. But the good news is that it doesn't take strenuous exercise. So there's a sweet spot. And what research is suggesting is that when you get to a point where exercise becomes stressful, then that's where you start to not gain as many benefits from it. Let's see what other questions we have here.
So another question we have is, are muscle memories associated with motor cortex? And so like the data from HM showed, clearly learning a new skill or something like a muscle memory is independent from the hippocampus, and the motor cortex is definitely involved in that process. Another brain region that's involved with it is called the basal ganglia. That's similar how the amygdala is involved in regulating emotion. The basal ganglia is involved in regulating kind of habit formation and how you're essentially controlling your movements. And so that brain region, the basal ganglia, connects very strongly with the motor cortex to help drive and strengthen these muscle memories. So something like Guitar Hero, really similar to the mirror drawing task, where the first time you played Guitar Hero, you can struggle a lot. It's really hard to keep up with the pace of the music. But as you practice more and more, you get better at it. So I think we have another question here. I'm going to pass it over to Rianne.
Hi, everyone. So we have another question asking, how can we reinforce what we learn each day? So kind of asking more, how do we better kind of improve our memory with certain types of information? Well, one thing you can do is kind of practice and keep testing yourself with these different types of activities, you know, trying to better improve in a certain area, make some type of memory. And this really kind of speaks to how I was talking about those brain cells, and really how if you keep doing some sort of task or trying to remember some sort of type of thing, you can actually make stronger connections with certain cells that are important for these types of memories or these types of activities. So we're going to start looking for a couple of other questions next.
Somebody asked us to place the resources page back up for you, so we've done that. Let's see what other questions we have.
So we got another question about kind of strengthening memory and strengthening recall. Specifically, this one is how to strengthen recall as one ages. So that's a big line of research like Manuela was talking about. We're still in the early days of figuring out the best ways to create long-lasting changes to memory. But this does seem like the strongest piece of data we have so far is exercising not just when you want to remember something, but exercising throughout your life will kind of set up a nice baseline for when you start aging to strengthen memory. So start keeping a good exercise habits now so that when your memory does start to decline, you actually have the extra boost of already having exercise habits to support that memory.
One other question is, has there ever been another patient like HM? So a patient that, or do we have any now that don't have their hippocampus, or do we still have to do that surgery now? And the answer is that we try nowadays to remove one hippocampus or one medial temporal lobe or the other. It is rare that they're both removed because now we understand the huge impact that that has on an individual if we remove both of the medial temporal lobes. So there are people nowadays who still have to have surgery. However, we typically remove one or the other, and we try to, not we, surgeons, try to be more specific about which areas they remove and removing only the areas that are absolutely necessary. Let's see what other questions we have here. So one question is about some memory tricks. So there are lots of memory tricks that are out there, but they all kind of have to do with this idea of elaborating the information. So if you have a list of words, for example, that you're trying to remember, instead of just trying to remember the words, saying the words out loud sometimes helps. Reading them out loud to somebody else, making connections between the words, making stories. So if you're thinking, for example, about a list where the first word is window and then the second word is dog, and you're trying to remember those words, you can create a story where the dog is looking out the window or something like that. Anything that elaborates that memory is going to make that memory stronger. There's also something else called the testing effect, which is a really neat effect that a lot of teachers are really interested in and is important for education. And the testing effect is the idea that if you have two sets of information, or one set of information that you're trying to remember, and you split up the group. So let's say you have a class that's trying to remember a passage, and half of the class gets to study the passage by just reading the passage, and the other half of the class gets to study the passage by taking a test on the passage. When you look at their memory later and actually do that final test, the group that was tested instead of just got to read the passage actually does better. So the idea here is that asking yourself or testing yourself during study actually improves your memory more than just reading the information. Hopefully that makes sense. And there's a lot of information. If you Google even the testing effect, you'll be able to find lots of information on that. And there's still research trying to understand, even though this was discovered a while ago, there's still research trying to understand why that is. Is it a modulation from the amygdala, or what's going on there? Why is it that we are able to remember it better if we test ourselves? All right, I'm going to let Alberto take the next question.
Hi, so we have a question about what happens to kind of these memory networks during dreaming stages of sleep. So for a little bit of background, in case people don't know, there are different phases of sleep, the sleep cycle. So when you kind of wake up groggy in the morning, it's probably because you got woken up, you got jarred awake during one of the deep phases called REM. When you wake up refreshed, it's when you kind of were allowed to come back to the top of the cycle when you're barely drifting asleep, and so you feel less groggy. But it's during that deep phase of sleep called rapid eye movement where the brain activity actually looks very similar to the activity of the brain while it's awake, which is really strange because you would think the deeper you're sleeping, the less like an active brain you would think it looks like. But it turns out that during it, well, this is still early phases in the research, but it seems like during that rapid eye movement, so when you're dreaming, the brain is actually replaying the information that it deems important. And so while it's replaying that, it's essentially resending those signals to try to strengthen essentially what are those memories that happened. And so things that happen to you during the day that you deem important or that the brain has deemed important, it's going to try to replay and strengthen. So that seems to be part of why sleep is one of the best enhancers of memory, because it allows your brain to essentially decide what's worth keeping and what's worth getting rid of, and then file it away into your actual long-term memory.
So we have another question here asking whether Alzheimer's is dementia. And so dementia is actually just an umbrella term, and Alzheimer's is a type of dementia, but there are also other types of dementia, such as Parkinson's disease, Huntington's disease, vascular dementia, and we're still trying to really understand the differences of how these different diseases kind of begin and how we're able to treat them differently.
So one of the questions is, how early in life does a person remember? And this is a really tricky one because some people will say, I remember being a baby, and I remember when I was a baby, my mom took me to Disneyland. But it's very hard to tell later whether that person is actually remembering that event or whether they're remembering the hundreds of times that their mother has told the world that she took this child to Disneyland when the child was a baby. And so, you know, memory is not a video recording of our lives. So every experience that we have after the event changes that memory. And that is not just for childhood, that's always. And so everybody is going to have a different age when they remember have their first memory. One really neat thing that we've started studying here at UC Irvine is people who can remember a lot. There are people in the world called highly superior. They have highly superior autobiographical memory, and they remember very specific details about many, many days of their lives. So you'll be able to ask them, for example, you know, what were you wearing on September 14th, 1984, and they'll be able to tell you. They'll tell you what they had for breakfast, and that's something that we are studying now to try to understand kind of on the other side of on the other side of the spectrum from things like memory loss is this superior memory. And if we can understand that better, you know, maybe we'll be able to understand how the brain learns in general and remembers in general better.
And so one thing to add about what Manuela was saying about the humans with superior autobiographical memory is, while we're all thinking it would be great to have that kind of memory, I'll never have to study for a test, I'll never have to worry about where things are in the grocery store, I'll know where everything is, but the kind of double-edged sword, the downside of that kind of strong memory is, like Rhiannon said earlier, some memories are tied to, or I think I talked about this, how some memories are tied to emotions. And so essentially, if you have a strong memory and it's tied to an emotion, in these individuals with a superior autobiographical memory, they also feel the intensity of that emotion as if it happened yesterday. And so that seems to be the downside of these strong memories. So I think we have a few other questions that we might get to. We're getting close to time.
Let's see here.
So we have a question about, is there a way to prevent Alzheimer's disease? And that is a large portion of memory research right now, trying to prevent essentially the progression of Alzheimer's disease. And right now, there's a lot of therapeutics out there. And it seems like the best way to prevent Alzheimer's disease is actually early detection. So a lot of research has started to come out and show that by the time an individual is symptomatic, it's essentially the fire is too big to put out. And so a lot of research is trying to develop early life biomarkers of somebody who may potentially develop Alzheimer's disease to prevent the fire from starting in the first place.
Let's see here if we can take a couple more questions. Trying to see if there are any questions that we haven't answered already. So it looks like we're all set. It doesn't look like we have any other questions that are on here that I can see. If you do have any more questions after the webinar, you're welcome to submit them below. Thank you so much for watching this webinar today. After we close, a short survey will come up. We encourage and appreciate your feedback so that we can continue to improve BrainFacts.org webinars. Thanks again. We really enjoyed this, and we look forward to you being part of future events.